So hello everybody and welcome to the ESDR Kitchen. I'm Eli Sprecher from the Department of Dermatology at the Tel Aviv Medical Center in Israel. And on behalf of the ESDR board, I'd like to welcome you all to this new sweet and sour episode of the ESDR Kitchen. I'd like to remind you that all of the ESDR Kitchen program, as well as all previous episodes, are available on our website. As you must now know, the sweet and sour episodes usually involve two discussants, which are addressing somehow controversial or unsolved issue. And today we are particularly pleased to welcome two well-known and world-renowned speakers, Professor Jakub Tolar and Professor John McGrath, who will discuss being able to cure EB or treat, but not cure EB. That's the question. So a few introductory words about each of our two distinguished speakers of today. Jakub Tolar is the Dean of the University of Minnesota Medical School and a distinguished Knight Professor in the Department of Pediatrics, Blood and Marrow Transplant and Cellular Therapy. He's also Vice President for Clinical Affairs at the University of Minnesota. He's an internationally recognized physician and researcher. He's very well known for his work on patients with recessive dystrophic EB. His research is focused on using hematopoietic stem cell transplant as a treatment for rare genetic disorders. He's originally from the Czech Republic. He received his MD in Prague at the Charles University. And in 1992, he came to the University of Minnesota, where he completed his PhD in molecular, cellular, and developmental biology and genetics. John McGrath is the academic head of St. John's Institute of Dermatology in London, where he also ran the genetic skin disease group. He also married and held chair in cutaneous medicine at King's College London, and is honorary consultant dermatologist to the Guy and St. Thomas NHS Foundation Trust. He has lead roles in delivering experimental medicine and in running several national rare skin disease projects in the UK. His clinical and research work focuses on the diagnosis of inherited skin diseases and the development of novel treatment for rare skin diseases. Now, before starting, I'd like to remind you that you can forward your questions through the chat or to the Q&A functions. We will have 10 minutes at the end of this of John's and Jakub's talk to discuss their topic. So John and Jakub, thank you on behalf of all of us for leading the show today, and the floor is yours.
Thank you very much, Eli, and good afternoon to you all. My adversary, Professor Tolar, are you ready to debate?
Oh, I am absolutely ready. I just want to say that I'm not at all intimidated by the fact that my honorable opponent is a proud product of the British educational system and has probably watched more debates than I ever have watched baseball games.
Yes.
That's not saying a lot, because I don't think I've actually watched baseball.
You're always a good talker. Let's get down to business, and here we go. So here is the title of our debate, very sort of Shakespearean, well, to some extent. But this is the scenario. These are patients with genetic skin diseases, this genetically and clinically heterogeneous group of disorders. And here is EB, equally clinically heterogeneous, and a huge number of genes that are involved in the diversity of phenotypes that you see presented here. And that's going to be relevant to our discussion about treatment. Now, when it comes to EB, a very quick reminder for some of you, most of the pathology in EB is somewhere near the dermal epidermal juncture, which for those new to the topic, it's a complicated macromolecular adhesion zone. So that little blue box there, just 200 nanometers across or so, is packed full of these sticky proteins that hold your epidermis to your underlying dermis. So you have this jigsaw, this sticky macromolecular network that prevents blistering. Now in EB, we think of those various proteins as a series of interactive protein rings. And the pathobiology of EB is to have mutations, mostly loss of function mutations, in a gene encoding one of those proteins, and that breaks the ring at various levels depending where it is. And where the ring is broken, you get the blister. And so here is the blister, and here magnified is the molecular pathology across those different types of EB, showing dermal epidermal junction, intercellular adhesion defects, and some intracellular abnormalities as well. Complex multiple pathologies underlie different forms of EB. And I think I would argue that's going to be relevant to our direction of travel over treatment. And the direction of travel over treatment, people think genetic disease, gene fix. Well, what you can see here is that the umbrella term EB covers 40 different genes. So that's going to be relevant when we think about the strategy to try and help this community of patients. And also we need to think about this burden of disease and unmet need. Wound healing is poor and slow. Patients suffer from incredible pain and itch. And so any of our therapeutic ambition needs to be able to address all of these particular aspects. And so here is a patient with one form of EB, with dystrophic EB. And the central tenet for our debate today is this. On the one hand, you could think about targeting the primary pathology that causes that weakness, whether you fix it with genes or cells or proteins that are specifically targeting that pathology. Or you could think about, well, let me survey the damage that's been done in the skin, and I should focus my therapeutic strategy on dealing with that very extensive secondary damage, which is mostly inflammatory. So here to put forward the debate is Jakub Tolar, who is going to say about that primary fix, and me, who is going to follow on from Jakub and just give you a brief summary of targeting inflammation. Because there's a lot out there. This is our buffet board of treatments, of genes and cells and proteins and various other things. And so Jakub is going to cherry pick a few primary fixes, and then I'm going to mop up what's left on the buffet board and give you the true morsels that will give you the diet of choice when it comes to helping patients with EB. So Jakub, I'll hand over to you to talk through your primary fix for patients with EB.
Thank you, John. I admit that I'm somewhat intimidated debating someone who I consider a mentor, but there's a saving grace here. I believe that my argument, striving for the cure, contains his argument that it's not possible and we should strive for more, as he says, addressing all aspects, primary fix treatments. And I should add that I personally know of no physician or researcher who, in trying to help a patient, is truly ready to settle for less than the complete eradication of suffering and the disorder itself. In other words, a cure. So we start with the gene therapy. This is the best case scenario, intermediate junctional LAMB3 epidermolysis bullosa. Phenomenal work by Professor De Luca and his team, and this Holo-7 approach being now expanded to collagen 7 and collagen 17 forms of EB. These are early days, but we are on the phenomenal example that defines the field. Click. Other trials out there are focusing on keratinocytes, fibroblasts, skin equivalents that have been modified to provide that all-important collagen or laminin. And again, you will associate it with number of people, but that ring around Palo Alto, Flain, Kavari, Marankovic, and Oro has been defining some of that territory along with Dr. Hovnanian and others. Click. The topical gene therapy is answering a need that the patients want, which is we want healed wounds, and the wounds need that that treatment on the target, if you will. And the topical collagen 7 type 7 collagen gene therapy with on the backbone of herpes virus type 1 attracted a lot of attention appropriately. Click. The latest data in this double-blinded, randomized, inter-placebo-controlled trial has shown that there is a significant difference in number of the patients. And again, early days, but it provides some hope for the next round of that. Click. We have focused on the high tech mostly, but not everything is high tech. Gentamicin, which is an antibiotic, of course, has been picked up by the brilliant idea of Dr. Woodley and Chen, and looked at a tool that can read through some of the mutations in the type 7 collagen, again, showing the first ability to restore some of the functionality of the broken skin. Click. The natural gene therapy has been intriguing. It has happened, of course, not just in genodermatoses, but outside of that in bone marrow failure syndromes, Fanconi anemia, dyskeratosis congenita, and others, and is a way of getting the skin restored with the tools that have happened initially in the body itself. So you can have, and these data are from the late Marcel Leonkrant's lab, you can have translatable benefit mediated by the grafting of the mosaic skin. Cliff? This led to a commercial level in Japan on the commercial level to ability to restore and expand the revertant skin and use it as a tool of therapy. Click. The RNA and gene editing, there's plenty to do, and since that has been initiated some years ago and I got a Nobel Prize, the first, to my knowledge, shared by two women, it has evolved into the prime editing and base editing, which does not require the double-stranded breaks or donor DNA templates and make it much more safer and much more on target. Click. The protein therapy is the maneuver whereby lets us break it down and deliver the missing protein and doesn't get much more targeted at the cause as that. And about 3 years ago, this trial started and results are probably imminent. Click. The stem cell therapy I introduced to you with the first slide in my section from the Professor De Luca, but our group and several others have attempted to restore the functionality of the external and internal wounds in epidermolysis bullosa by using of bone marrow transplantation. This is the ultimate systemic therapy. This is the combination of the both causative and anti-inflammatory and immune tolerogenic therapy, and again, a platform on which we can build. I turn it back to Dr. McGrath to finish the other part of the equation. John?
Thank you very much, Shaker. Very eloquent indeed. So we've heard a lot about this focusing on the primary pathology, but now it's time to park Dr. Tolar for a moment and focus just on what really may be for patients the better approach to go, which is to focus on that secondary pathology, which is mostly inflammation. Because when one looks at dystrophic EB, for example, it's not just a surface disease. You have this raging, uncontrollable torrent of inflammation, scarring, and pro-cancerous development going on beneath the surface. And we really need to be able to target that rather than just putting a superficial paper layer of ex vivo gene therapy onto the patients. And EB is often a systemic disease as well. So whatever we're going to do, we're going to need to target that multi-organ dysfunction as systemic disease. And also we've got to really think what patients want fixing. And when you ask patients what they want, they don't say, cure me. They want to have an improved quality of life. They want better wound healing. They want reduced pain. They want reduced itching. And so I would argue that for these high-tech approaches, cell therapy, one of the most advantageous approaches for dystrophic EB, may be to adopt what we could call the fire extinguisher approach, an anti-inflammatory immunomodulatory approach, using something like intravenous allogeneic mesenchymal stromal cells. Here you can see a before and after change in quality of life, reduced pain, reduced itching. It's what the patients want. And we know roughly how those MSCs work in terms of that anti-inflammatory soup of stuff that then calms down the skin and improves quality of life. And that cell therapy is advancing now to more specific and better immunomodulation and anti-inflammatory approaches, whether it's using ABCD5 cells or new cells. These are moving forward nicely. And of course, the great work from Katsuto Tomai's lab about mobilizing endogenous stem cells with a recombinant peptide to part of the high mobility group WOX1 protein is another important generic form of anti-inflammatory benefit for many patients. And so this idea of targeting inflammation can help both the patient and also the disease pathobiology and the progress of disease. So we really need to get in there and target inflammation, and it could reap many benefits for patients. But look at these sort of approaches, biologics, which all you dermatologists know are out there for inflammatory diseases. They can also help genodermatoses and conditions like EB. So you can see dupilumab in EB pruriginosa. Reducing the itch, that major symptom of patients using a JAK inhibitor, it can be very helpful for many of these patients. It's important that we give access to these drugs to patients with EB, but that may be another debate. And when we get a little bit more scientific, our colleagues in Italy looked at these two identical twins with different forms of recessive dystrophic EB and worked out the differences. Why was the individual on the right less inflamed? It turns out there were different cytokine and growth factor factors present there, but also decorin, an extracellular matrix protein which inhibits TGF-beta, was increased, so the TGF-beta was reduced in the good twins' skin. And that led to mouse models, RDEB mouse models, overexpressing decorin, showing reduced fibrosis, and has then led to clinical trials where we try to target disease progression, reducing fibrosis, improving quality of life for patients. So there are all these multiple phenotype modifying trials out there for patients with EB. And I would argue that this is probably the best way to go for patients trying to help improve quality of life. And so Jakub, this is a figure from one of Jakub's own reviews where he presents the idea that combinations of therapy, i.e. not a single approach to correction, is going to help the patients. But I would argue that what he really needs as well is a rain shower of anti-inflammatory therapies to put out the fire, the inflammation that is driving EB and many other genodermatoses. And the final pitch would be a socialist one, a numerical one. There are about 500,000 people in the world who've got EB. And I would argue that Jakub's approach to gene therapy is all very high tech, and it's designed just to benefit those very few patients who are above the surface, the tip of the iceberg. What about the burden of disease and the unmet need for the vast majority below the surface? Surely it would be much better as a global strategy, not to try and cure EB, but to improve quality of life through targeting inflammation, improving wound healing, reducing pain and itch. So here are the community of patients with a desperate need. Maybe we could agree that there will be a combination of therapy, but I think in terms of the balance and the emphasis, I would like to argue that perhaps we should focus on inflammation and treating EB rather than directly trying to cure EB. And with that, I will rest my case in this debate. Thank you very much.
That was brilliant, Professor McGrath.
And likewise,
I don't know whether it's more an advantage or disadvantage knowing that my honorable opponent agrees with me.
Exactly. Although one could argue that much of your case was somewhat specious, Jakub, for my, but never mind. Eli, let's pass it back to our moderator.
Yes, so thank you both for really a brilliant couple of talks. And I think that this is something, of course, which has to do with a specific disease, EB, but as a team, it can be also expanded to many more disorders that we are trying to tackle. And the question is also always whether to target the root of the disease pathogenesis or to be more efficient at alleviating the suffering of our patients and targeting the end results. So we don't have too many questions. You guys are, I guess, a bit intimidating, but so I will try.
It's only Professor McGrath, but Eli, to your point, I would say that that's exactly why we have a treatment for leukemia, my day job. That's exactly why we have a treatment for cardiac disease. That's why we don't even know, my residents and fellows sometimes cannot recognize infectious diseases that were lethal just 50 or 60 years ago. And I would argue that if we aim for a lower bar, that is the highest goal we reach. It's like an archer. If you want to hit the target, you aim a little above that. And if we reach for the highest bar, we may leap over the lower bars more easily, even if our highest ambition is not successful.
I would argue, Jakub, though, the challenge we've got now is that there have been such great advances for patients with skin diseases like psoriasis and eczema, much of which has been presented at ESDR meetings. But we have biologics, we have JAK inhibitors. And for patients with genodermatoses and EB, it's like being locked outside a sweet shop. Those goodies exist inside the sweet shop, and we don't have access to them. If you have eczema and psoriasis, you can have access to these. You fail a systemic treatment and you get your dupilumab or other form of biologic or JAK inhibitor and so on. We can't even get our patients with EB these treatments very easily. We need to unlock the door, and I'm happy to be the shopkeeper that can hand out some of these biologics to patients in proper clinical trials, but we need to get access to them. And I think once we get access to them, we will start to benefit the many rather than the few. And that should be the emphasis of where we go. I fully agree. It would be great to combine therapies. You and I think, both agree that all of this combination therapies, either simultaneously, concurrently, or at different stages in life, is the way to go for EB and many other diseases. So, but I think just in terms of what I would like to prioritize at the moment, it's maximizing use of existing resources and stop denying patients with EB access to potentially useful, life-enhancing medications. That would be, I could benefit these patients within a week. You could benefit some of your patients within 5 years. That may be the different emphasis of where we ought to go with this debate.
That's very well said, John. And I would add to this that if you are the shopkeeper, as you say, if you are the chef in this ESDR kitchen, all is well, because you will not only know that borrowing from other fields, oncology, rheumatology comes to mind immediately, is exactly what needs to be done, and exactly that needs to be presented in your kitchen, along, of course, with tea and crumpets and Eton mess, maki dripping, periwinkles, and other brilliant accomplishments of British cuisine.
Well, there we are. We should hand back to our maitre d' before we get too carried away, Jacob.
Absolutely. So a more down-to-earth question, and actually something which is, I think, in starting to be used in both contexts. So in terms of cell therapy, John, can you enlighten us about potential use of exosomes, which are kind of the new cell therapy generation? Any prospects of using those in the context of EB or other genetic skin diseases?
Yes, I think you're right. I mean, we know that using a whole cell, an MSC cell, is very useful. And any protagonists of cell therapy have often thought that the future must be reductionist. You must be able to find exactly what it is within a cell that will help the treatment that you give to the patient. And so one of those magic ingredients potentially are exosomes, these little vesicles, which we know contain more than 800 different proteins and RNAs and microRNAs, and which can potentially be harvested from an industrial source and then packaged in a relatively stable form, and then used as fire extinguishers to put out various inflammatory therapies. And maybe they will be the corticosteroids of this current decade. The challenge, of course, when it comes to cell therapy for disease is that nobody has quite been able to recapitulate the benefits of giving an entire cell at the moment. There is always an element of grandma's chicken soup when it comes to giving the whole cell, whereas if you try and break it down into fine bits of chicken or certain bits of stock in terms of exosomes, it doesn't always work. But I think you're right. It would be lovely to have this generic form of non-inflammatory, anti-inflammatory indeed therapy in the form of exosomes derived from MSCs or indeed other cells. So I'm sure that will be a direction of travel that many people wish to pursue. But I'm sure Jacob has an opinion about exosomes in the context of leukemia therapies and some of those other immunomodulation that he's involved with. Jacob?
Thank you, John. Yes, but I'll restrain myself and comment on the EB part of this, because our lab has in fact collaborated with a group in Singapore some years ago and tested these exosomes that were derived at that time from immortalized human cells, not necessarily mesenchymal stromal cells. And we have found benefit of this. The challenge in the murine model, that is, the challenge has been, of course, the translation into the clinical realm, and the instability of the product has been one of the shortcomings of this, because the goal there was the off-shelf, sort of immunologically blind therapy, you know, that you can essentially use this for anyone with any kind of matching, in a kind of rejection, and in a dose. So the dose has been the second concern, the effective dose, that is, you know, on the side of delivery. I would think that the targeted delivery, and again, I apologize for agreeing with you now, I'm not supposed to do that, but I do think that the local therapy that's aided by, say, nanoneedles, or, you know, some kind of a gel, you know, or the liposome base, you know, the exosomes, of course, have a lipid membrane, may be the right way to initiate some of that combination therapy that you presented. And yes, of course, this needs to be done in stages that has the anti-inflammatory, and I would argue the immune tolerogenic phase, before you come with these architectural bodybuilding pieces that will restore the skin and the internal membranes, essentially.
We have actually, as a kind of follow-up to what you were just discussing, a question from Chris Griffith, and he's asking which cytokines would you target with biologics, and he's also asking whether MSCs are actually not better in terms of a broader range of targeting. So
I will ask
versus
my honorable opponent to comment on the cytokines, and I'll take the MSCs. John?
So certainly in terms of with the advances in omic technologies and single cell transcriptomics and so on, we're now starting to get some profiling of what's going on in RDEB skin. And we've even got heat maps before and after treatment with various biologics. We've seen that for some itchy forms of EB using dupilumab. Beforehand, you can show some abnormalities of Th2 cytokines, and you can show the restoration to almost normal skin in the transcriptomic profile after giving something like dupilumab. We're also finding in EB that just like it is in about a third of all genodermatoses, there seems to be some Th17 abnormalities as well. And as a blind treatment, it almost feels that anti-IL-17 therapy can probably benefit at least 30% of patients with genodermatoses. And there is before and after transcriptomic evidence to show that that may be a good way to go. So the emphasis at the moment is around Th2 cytokines and IL-17 and its associated pathways. And it's preliminary data at the moment. It's small numbers, but that's kind of the supportive data at the moment.
I'm going to add in 60 seconds that the mesenchymal stromal cells have been another tool that has been borrowed from other fields. We have first used them clinically in the bone marrow transplant for an autoimmune complication of allogeneic transplant called graft versus host disease. These are bone marrow derived cells that have their main course of action or essential action in T cell modulation, in the trafficking on the external and internal membranes. There's an additional class that has been derived from the skin and has ABCB5 marker on the top, and are again skin-derived mesenchymal stromal cells, and their mechanism of action is slightly different because it's the polarization of the macrophages and monocytes, which lead to the change from the chronic angry wound to the acute and then healed one. So there is a combinatorial opportunity here again, and I would argue that it is in the middle of our debate is what I would call radical incrementalism. You know, where you go and quickly sequence things that are rational, that are supported by evidence, partial perhaps, and accept that the risk of not knowing how this is going to end is a feature of the system, not a bug.
I would argue also, Jacob, that many of those incremental advances around some of those cells that you talked about are still work in progress and maybe not worthy of a Michelin star just yet. They may turn out to be very tasty, but I think it's still stuck in the kitchen.
Now I have a challenging question for each of you, and you have 30 seconds each to answer it. And the question is coming from Sheila McSweeney. She's asking about the difficulties of running clinical trials in the context of rare genetic diseases, of course, including skin genetic diseases. So, John, you start.
Certainly. Thank you very much, Sheila. So the challenges are there for patients. you need to be able to recognize and access your patient group. They're relatively small numbers. So any trial you're going to be able to design for patients, your N number for power is going to be very limited. So defining your endpoints, primary endpoints, and probably having a collection of secondary endpoints is going to be the way forward. And then you've got to see what's available, what you can get access to, what you can get permission to use. So much of what we would like to use, we don't have access to at the moment, but it requires a lot of stamina, a lot of effort, a lot of determination to keep going to try and help patients with this unmet need. So the logistics are there in terms of challenges, and that's where I would focus in trying to help these patients. Jacob?
My 30 seconds as the Athena emerges from John's head. As you heard, I would add to this that the mindset is incorrect. First, the challenges of the clinical trials are a part of doing this. The problems with them are a parameter of their function. So if you want to do something meaningful for the patients, you better get to the position that you can actually go through the adversity of submitting and running clinical trial. The second thing is that the mindset is incorrect also in the way that we are biased towards these many hundreds, many thousand kind of clinical trials with very, very small variation. Pharma likes that stuff in the progress. That's probably incorrect, because I've argued a decade ago of the IND with N of 1 and the precision medicine that we now are seeing, with the genomics and everything else, is arguing for the same thing. You tailor the therapy, whether it's gene therapy, cell therapy, or other protein therapy. You tailor the inflammatory, the response, everything to that single patient, and it's entirely doable, especially with artificial intelligence and machine learning and decision algorithms that are available to us, again, from the other fields than dermatology. Ali?
So I think that our debate comes to its end. So I'd like to very, very much thank you both for an inspiring debate. And I'd like to remind everybody, we will meet again for our next ESDR Kitchen on February the 23rd, a recipe book, a new episode on mouse models by Nicole Ward and Rupesh Singh. Thank you all for having been with us, and goodbye till the next time. Bye-bye.